All right, let's talk about the neuromuscular junction. So the neuromuscular junction is where a motor neuron makes contact with a muscle fiber. Each muscle fiber is only going to have contact with one motor neuron. One motor neuron, though, can have contact with multiple muscle fibers. And this is what's going to allow for smooth, even contraction. Now, where the motor neuron encounters the muscle fiber, the end of that motor neuron is going to be referred to as the axon terminals, or the ends of that particular motor neuron branch where it makes contact. A nerve can have multiple axon terminals. This gets into a little bit of a conversation we haven't had yet, talking about the nervous system. So we'll explain that in more detail when we get there. But axon terminals are the end of an axon. A nerve can have multiple axon terminals. So the axon terminal is the end of the motor neuron. On the muscle fiber side, it's going to have a motor end plate, and that is going to be where the receptors for the neurotransmitters that are going to be released from the axon terminals, those receptors are going to be located. It's a specialized region adjacent to the axon terminals, and they're going to contain the receptors so that this communication can occur. A motor unit is going to refer to the motor neuron and all of the muscle fibers to which it is connected to. So all of the muscle fibers that it talks to. That could be one, or that could be half a dozen to a dozen to a couple hundred, depending upon the motor neuron and the muscle involved. As I mentioned, they can be really small. These would be found in like the larynx and the eyes, or they could be large, upwards of thousands, biceps, gastrocnemius, lower back muscles. When we get into skeletal muscle microanatomy, so we're getting into the individual cells, these are going to be have a diameter of upwards of 100 micrometers and be as long as 30 centimeters. All depends upon the muscle that they are located at. They are this large since a single skeletal muscle is the result of the fusion of hundreds of embryonic precursor cells called myoblasts, and this is also why they're multinucleated. A single cell is actually composed of a bunch of cells smushed together. Now, when we have cells that are a fusion of multiple cells, we actually refer to this as a syncytium. Don't stress about that. All right, getting inside of the skeletal muscle, the plasma membrane of the skeletal muscle is referred to as the sarcolemma. So instead of calling it a cell wall membrane or a plasma membrane, we refer to it as a sarcolemma. It has its own special name. The cytoplasm, so the fluid inside of that membrane, also has its own name. Instead of referring to it as cytoplasm, we do refer to it as sarcoplasm. Now, the endoplasmic reticulum, specifically the smooth endoplasmic reticulum that is found in muscle cells, skeletal muscle cells, also has its own name. We refer to that as sarcoplasmic reticulum instead of endoplasmic reticulum. So, a couple of different names that we do need to be aware of. Now, the sarcolemma is going to have invaginations or pouchings that penetrate through the cell, and this is going to be referred to as a transverse tubule or T-tubule. These T-tubules are going to have on either side of them some structures, and that's all going to come into play when we talk about contractions. Other things to note, a skeletal muscle cell is going to have a ton of mitochondria. And if you remember what mitochondria does and what your muscle cells do, hopefully that makes sense to you as to why. If you don't remember that mitochondria are involved with energy production, and your skeletal muscles are energy hogs. So they're going to need a lot of energy. We're also going to have things such as glycogen granules to provide glucose as basically storage pouches on demand. We'll have things called myofibrils, and that sarcoplasmic reticulum. Now that sarcoplasmic reticulum is a variation on smooth endoplasmic reticulum, as I mentioned, only instead of making fatty acids, it's going to store calcium. This is going to be important for muscle contraction, and we'll explain in the next audio why that is. We're going to have a loose network of this sarcoplasmic reticulum throughout all of the myofibrils in a particular muscle fiber. And again, we'll explain why this is important when we talk in the next audio. What is a myofibril? A myofibril is a bundle of rod-like structures that extend the length of the cell. They're going to be long protein structures called myofilaments, and their actions are what are going to give give the muscle, I should say, the ability to contract. These myofibrils or filaments are going to be divided into two categories: thick filaments and thin filaments. Here's our artistic rendering again. Sarcolemma, sarcoplasmic reticulum, mitochondria, myofibrils, thick filaments, thin filaments. All right. Now, thick filaments are going to be referred to as myosin. They are made up of the protein myosin, and they kind of resemble two golf heads that have their shafts that have been twisted around each other. A single section of a thick filament is going to have hundreds of myosin molecules. Approximately 300. This is all going to depend upon where we find them. We're going to have a lot of these guys. And this is what they look like. They are literally, there's going to be hundreds of these heads, and they're going to kind of circle around the entire filament. Again, why is going to come into play when we talk about contractions. Now, its opposites, what is going to come in contact with during the contraction portion of this conversation, is going to be the thin filament. Thin filament is known as actin, and it's actually made up of three different types of protein: actin, tropomyosin, and troponin, but we simply refer to it as actin. Actin is going to actually be a double helical strand, long double helical strand of actin molecules. On top of the actin molecules, we're going to have the troponin and the tropomyosin. Now, this typically, the actin has a very bead type appearance, globular appearance, so it kind of looks like a twisted strand of beads. On each actin protein will be a myosin binding site. This is, again, going to be important when we talk about contraction. Loosely wrapped around the actin helix and covering those myosin binding sites is going to be the filamentous protein tropomyosin. On the tropomyosin and connecting the tropomyosin to the actin is going to be a trio of proteins known as the troponin complex. These are going to be structures that you do want to remember, and the fact that actin has the myosin binding site, because they all come into play when we talk about how your muscles contract, the skeletal muscles specifically. All right, myofibrils. Each myofibril is going to be made up of thousands of repeating individual units known as sarcomeres. Sarcomeres are the functional unit of the muscle cell. This right down here is an artistic rendering of a sarcomere. Each sarcomere is a ordered arrangement of thick, thin filaments. In this artistic rendering, the thick filaments are these purple with the heads, and the thin filaments are this pink. And typically you will find that the thick filaments will come in contact with multiple thin filaments. And the thick and thin filaments are going to overlap. If they don't overlap, we cannot have contractions. There are various zones and labels of areas of a sarcomere. The two flanking protein structures are known as Z lines or Z discs. These are going to denote the ends of the sarcomere. The portion of the sarcomere where we find the thick filament is referred to as the A band. The portion that does not have any thick filament is referred to as the I band. It will appear very light under a microscope and only contains thin filament. The H band or H zone is the portion of thick filament that is not overlapping with thin filament. We're also going to have an M line, which is the centermost line or the zone where no contraction can happen. There's some other zones and bands, but we're really not going to focus on those. All right. Going back to our T-tubules and sarcoplasmic reticulum. So T-tubule is a pouching out of the sarcolemma, and it's going to extend all the way through to the other side. Off of each side of this is going to be what we refer to as lateral sacs. These lateral sacs are going to extend into the sarcoplasmic reticulum and are going to enable the flooding of calcium into the sarcoplasm, which will enable the contraction to occur. Now, these lateral sacs are also typically sometimes referred to as terminal cisternae, depending on the textbook that you're using. Now, each T-tubule is going to have two lateral sacs or terminal cisternae, and we refer to them as a triad. So when you're reading in a textbook about skeletal muscles, contractions, and you see the term triad, that is what it's referring to.